Here we are, AP Biology, Chapter 9. We are looking at heredity, genetics, inheritance, or the patterns of inheritance. This is the second video in our series. This video is going to focus on the law of independent assortment that was developed from Mendel in his experiments. If we're going to talk about Mendel, we're going to need to go back and review our first video. And so we'll look back at what Mendel was. Mendel was the Austrian monk who was doing experiments using pea plants. Specifically, he was crossing, to look for traits. So he looked at flower color, flower position, seed color, seed shapes, pod colors, and then lastly the stem length. And what he eventually found out as a result of looking at this is he came up with what is the 3-to-1 ratio. And the 3-to-1 ratio is 3 times it showed up the dominant trait, 1 time it came up the recessive trait. And how was it when he cross-pollinated those true breeding plants? And so we're going to look at that in our next slide here. So when we look at what Mendel was doing, let's focus on just one experiment that he did. And here he crossed the true breeding purple plant, which would have had the genotype capital P, and he crossed it with the true breeding white plant, which in this case would have been the lowercase p. And when he did that, what he produced was all purple offspring. We now know that all of those purple offspring were 1 capital P, 1 lowercase p. And how do we know that? Well, if we were to take this capital P, purple flower, and cross it with the lowercase p, white flower, what that Punnett square looks like when we do the cross is right here. So you can see we're crossing our lowercase p, which is right here. We're crossing our capital P, purple flower here. And when you combine those two alleles, what you end up with is big P, little p. And you end up with it big P, little p, or heterozygous in every single possible combination of creating a purple and the white flower. However, what does big P, little p's phenotype look like? They are going to look purple. But what Mendel found is that when he allowed this big P, little p flower to reproduce with itself, what happened was the recessive white flower showed up 25% of the time. Now, how does this happen? Well, if we were to look at a Punnett square for big P, little p, and we're crossing it with big P, little p, what happens is we can create an offspring that is homozygous dominant, big P. We can create a heterozygous offspring. We can create another heterozygous offspring, or we can create a homozygous recessive. Now, when we look at this and we say, what does this look like? These top ones are going to look purple. This one is going to look purple. And this last recessive one is going to be white in a ratio of 3 purple to 1 white. And this is exactly what happened for Mendel every single time he did his experiment over the 2 generations. And so as a result of that, he came up with the conclusions that organisms have 2 alleles or 2 versions of every single gene. And one of those alleles is dominant. In this case, this was our purple. And one is going to be recessive, which is our lowercase p. And then that organism is going to express the dominant phenotype. In this case, again, it's expressing that purple. Now, however, that organism can only pass on one of those alleles, and that is this Punnett square that we're looking at here. When they pass on, what happens is that those alleles separate. Therefore, during meiosis, we're segregating the alleles, creating a new offspring. And the new offspring, which is our F2 generation right here, could be purple, or in this case, there's the one that turned out white. And that's the law of segregation. So because of Mendel, we discovered alleles, we discovered dominant recessive, and we discovered the law of segregation explains how that recessive phenotype can show up in the next generation. Now, the other thing that Mendel discovered is called the law of independent assortment. And the law of independent assortment was only discovered when he looked at 2 characteristics at the same time. Now, these 2 characteristics, he was saying, okay, what are the odds that you would inherit purple flowers and yellow seeds. What are the odds that you would inherit yellow seeds and wrinkly seeds at the same time? By doing this, what happened is the Punnett square got bigger. And this is where we get into the law of independent assortment and looking at allele formation or gamete formation for 16 boxes. Now, what's going on here is we are going to be focusing on what are the odds that you'll pass on the yellow, smooth, and green, wrinkled. So now we're looking at all of these different traits. When Mendel did his experiment, so again, he cross-pollinated yellow, smooth, green, wrinkled. What he found was in the F1 generation, so here's our F1 generation right here, they all came out yellow smooth. So we know that yellow smooth is dominant. However, what happened when he allowed that generation to reproduce, to create the grandchildren, is he got 9 round smooth, yellow round. He got 3 green smooth round. He got 3 yellow wrinklies, and he got 1 green wrinkled. So how does that happen? Well, if we were to look at the law of independent assortment as it applies to the bigger picture, and we look at meiosis in more detail, what we can find is that as we go through meiosis, what's happening is that the alleles do not care where the other alleles are. So if you look at how the alleles line up here, you can see that the big R and the little r match up right here. But the big R does not care if it's on the left or it's on the right. And this matters because as we go through metaphase and anaphase, what happens is that the alleles on the left go to make one gamete and the alleles on the right go to make the second gamete. So what happens is we end up creating 4 different possibilities. Those 4 possibilities are the dominants get inherited together, the recessives get inherited together, maybe 1 recessive and 1 dominant, and again, the other combination, the other dominant, the other recessive allele. And that's as a result of meiosis and how it works in forming the gametes. Now, because of these 4 possibilities, what happens is we have to create a Punnett square that is much bigger. And the Punnett square we create is known as a dihybrid cross. So here you can see the 4 possibilities. The dominants were inherited together, the recessives were inherited together, 1 dominant, 1 recessive, excuse me, 1 recessive, 1 dominant. Now, as we go through this, what's going to happen is we'll talk about how do you build that dihybrid cross so that you can do the genetics and the possibility with it. Now, one of the real-life examples that currently exists is for Labrador retrievers or labs. It turns out that if you're looking to see, are they inherited together or separate, there is coat color, which you can be black or brown, and then there is blindness, whether you can see or you're blind. Now, what happened when they crossed the animals and they wanted to know is blindness, which is recessive, and chocolate coat color, which is also recessive. What happened when they crossed these animals is they created the same ratio that Mendel found, which is 9 dominant, 3 dominant for one and recessive for the other, 3 recessive for one, dominant for the other, and then 1 which is completely homozygous recessive for both traits. Now, that ratio gives, again, the indication that we understand meiosis and we're using the dihybrid cross. One of the things that happens with this dog or coat color is you'll see that they're saying this black coat, this black colored lab, they don't know what the second allele is. And that's because black is dominant. So even if you only have one of those alleles, you're going to look like the black coat color. So what happens is, how do you figure out if that dog is homozygous dominant for black, so capital B, or if it is capital B, lowercase b, which is heterozygous for the black coat color? Well, what you'll do is you'll do something called a test cross. Now, the test cross is used to determine the unknown genotypes, and here's how it works. if you have a black lab and you know it has at least one big B because it has the dominant color, but you don't know what the second is, you're going to cross it with a homozygous recessive. Because you're crossing it with a homozygous recessive, what happens is that by looking at the offspring, we can see what's possible. If you have homozygous dominant black lab and you cross it with a you cross it with the homozygous recessive, you're always going to make black labs. So if all the offspring come out black, you know that you have a homozygous dominant black lab. However, if you do the exact same cross, but the black lab is actually heterozygous, what happens is you're going to create some black labs, but you're also going to create some brown labs. So by looking at the offspring, we're able to determine the genotypes of the parents, even if it's not expressed in the phenotype. The whole thing about the genetics and the test crosses and Punnett squares is that we're actually looking at the rules of probability. We're looking at chance. And so when we talk about the chance, we're saying, what is the chances or odds that two things would happen together? And in order to calculate that, we're going to have to multiply. So when we say, what are the odds that you would become black fur, and we want to know if you're going to have blindness. Well, in order to do that, what we would do is determine the odds, so like 3/4, times 1/4. And then that would give us our rule of multiplication. However, what if we want to know that you could have this happen or this happen? So in this case, we want to know what are the odds that you would inherit black fur or you are going to be blind. So the odds of inheriting black fur in our case is 3 out of 4. Or, so we're going to add. The or makes the add. Or you're blind, which is a 1/4 chance. And so that's how we can use the rules of meiosis and probability of the Punnett squares to calculate multiplication or addition. Now, with this being said, I want to talk briefly about how do you set up a dihybrid cross. What we're going to do is we're going to use a method known as FOIL. So here is a heterozygous, we have the R, which we can say is round, and then we have the Y, which is yellow. So we're looking at a dihybrid cross, and we want to know if round equals big R, which is dominant, or we want to know if you're yellow, which is big Y, dominant. We want to say, what are the odds that you'll be round and you'll be yellow? Okay. How do we figure out how to set up the Punnett square? Because our Punnett square is going to have 4 possible alleles. Well, here I have outlined what is known as the FOIL method. So here is first, outside, inside, last. So first, you're going to take the first of the 2 different alleles. So here's our first. Big R, big Y. Oops, I copied the wrong arrow. I apologize. So big R, big Y. That's our first possible allele. I should make that very obvious, big Y. Next for FOIL, F-O, outside. So what we're going to do is go, whoa, and what do we have? Big R, little y. If we're to do the inside, little r to little y, little r to big Y, and then our last one is the last alleles for each set, so little r, little y. These are the 4 possible alleles that we'll then be adding to our dihybrid cross. And so here is a dihybrid cross that I've blown up. And you can see this is exactly how I set it up. So you'll do the FOIL method for each one of the parents in your Punnett square. However, you're going to fill it in the exact same way that we've seen. So big R, big Y, we're going to fill in big R, big Y, big R, big Y. Here we've created the little r, big Y, big R. So we're combining. What I want to show you is that the little r always goes after the dominant in this case. So we always write the dominant letter first. We always put the recessive letter last. Okay, here we are. That's the done. We are going to be talking or continuing on with Mendel's genetics or inheritance in our further videos.